fibroblasts are generally considered to grow better, more vigorously in culture. So we thought, what if we look at embryonic fibroblasts? And we chose the squirrel because the squirrel fibroblasts are the slowest proliferating in our assay. So we thought, what if we isolate squirrel fibroblasts and see how they do? Squirrel embryonic fibroblasts. So that was, of course, a rather challenging task because squirrels, in contrast to mice, they do not breed continuously throughout the year. So they have one breeding season in February, so we had to capture pregnant squirrel at the right gestational age, but we succeeded and isolated embryonic squirrel fibroblasts, and we were very pleased to see that at first these cells proliferated with the same rate as mouse cells. So this is a reference mouse, so these are adult mouse fibroblasts. But later on, after about population doubling 25, the proliferation rate slowed down and the curve became parallel to the adult fibroblast curve. Meaning that our conditions are okay, but there is certain mechanism that gets activated at this point, or perhaps when cells become more differentiated or more adult-like, that now starts to restrict cell proliferation. And I see some parallel with activation of other tumor suppressor mechanisms during development. For example, in human development, telomerase is very active in embryogenesis, but then it becomes progressively shut off later on. And so here, of course, it has nothing to do with telomerase, but there is a similar dynamics where this mechanism is not functional in embryogenesis when we need rapid cell proliferation, but it gets activated later on. And then we analyzed correlation between proliferation rate and maximum lifespan, and we see very strong negative correlation here, meaning that cells from longer-lived species proliferate slower in culture, and this is the data where we excluded those species that display duplicative senescence. So these are only the species that whose cells proliferate continuously, and there is no correlation to body mass. So now, to summarize, what we have found is a set of rules that sort of control evolution of tumor suppressor mechanisms depending on species body mass or lifespan. So here, if we look at animals with body mass and we estimate about greater than about 8 kilograms, we expect to find duplicative senescence. So among rodents, the examples are capybara, paca, American porcupine, and American beaver. And it seems that this rule is true also beyond rodents. So for example, for those species that have been analyzed, so mostly farm animals, like large animals like cow, sheep, and goat, they all display duplicative senescence similar to humans. And it also explains very well human versus mouse difference. So we display duplicative senescence not because we are longer lived, but we're just much larger than much larger animals than mice. So now if we look at species that are longer lived but small, so species with a lifespan greater than 10 years evolve those alternative anti-cancer mechanisms. We expect to observe slow proliferation rate in vitro. So now our model, to bring everything together. So if we have an ancestor here, and when species evolve larger body mass, the cancer risk goes up, and that's when species evolve repression of telomerase activity and duplicative senescence appears as an anti-cancer mechanism. So then, in species that evolve long lifespan but that are small body, their cells show slow proliferation rate in culture, and they evolve alternative telomere-independent mechanisms to protect themselves from cancer, because perhaps this cancer risk comes in a different stage of life. So here we are dealing with cancer risk during development, and here is something where they are subjected to increased cancer risk as they grow, as they live longer. And the last, we have species that are short-lived and small, and they just have fewer anti-cancer, fewer tumor suppressors, and humans fall into this group, and mice fall into this group. And of course, so this group is something that is sort of kind of uncharted territory and much less studied. So of course you would wonder, okay, what are those alternative tumor suppressor mechanisms? And yesterday, if you heard Andrei Sulanov's talk, he discussed naked mole rat, and this is a classical example of a small, long-lived rodent. naked mora doesn't display, the cells don't display replicative senescence, and it has a really novel mechanism that Andrei talked about, early contact inhibition, that seems to protect, to confer tumor resistance to naked mora cells. So now what about other rodents? So now we know, we have some understanding of those mechanisms in naked mora, and what I can tell you about, so we are looking of course at other species, so it seems that those alternative mechanisms are not the same across small long-lived rodents. For example, eastern gray squirrel cells do not display early contact inhibition. They can proliferate to high cell density on a plate, but still they proliferate very slowly. So perhaps those mechanisms are different, and our hope is that if we understand those mechanisms, they can somehow be applied for tumor prevention in humans. And finally, I would like to thank members of my group, so graduate students who participated in this, performed those experiments, Chris Hine, Jorge Asprua, and so also many undergraduates who worked very hard to produce those fibroblast growth curves. Of course, I would like to thank my lifetime collaborator, Andrei, and he performed also many of those experiments, because, I mean, unfortunately, I really don't get to do many experiments in the lab. I'm busy with some boring things like grant writing. And so, but now Andrei has obtained, his independent position, and so maybe soon I'm telling him, you won't have as much fun anymore doing experiments. And also our collaborators, evolutionary biologist David Presgraves, and Ken Catania, naked mora researcher, and our work is funded by NIH, Ellison Medical Foundation, and AFAR. And thank you all for showing up early in the morning.